I've added an extra reading assignment for this semester. I think it's a really good paper. I won't always do this, but I'd like to provide just kind of a short guideline. Now, before I do this, just a word. You hopefully have already realized this, but when I give assignments from the scientific literature, sometimes they can be a little bit dense, and I don't expect you to learn every single little bit or understand every part of the paper, but I think it's an important exercise, an important step to begin to a little bit of a higher understanding for these upper division courses. So don't feel overwhelmed if you don't get every little bit. The idea is that then in the videos, the lectures, the reading questions, you'll kind of get a guide for the important parts. And if you have trouble with any of the lecture material or with the reading questions, then maybe you need to go back in and review the paper in a little bit more depth, or bring it to one of the Zoom office hours and ask some more questions. This one we will do in a little bit more depth. I'll add some additional elements to the student learning outcome to also guide you through what I go through here. So I'm not going to provide a PowerPoint for this, but I will highlight I've already done that, highlight some of the important concepts here as I go through this paper. And so I'm just going to kind of walk you through it, hit some of those highlights. This is an important paper. It was fairly recent. It is a really good overview. Again, there's some technical stuff here, and you might feel a little lost in certain parts. But by and large, I think it's a very good overview of some of the recent understandings of the origin and history of animals, or the metazoa. Okay, so I've hit some highlights here in the abstract. Some of the very important ideas about animals are transition to multicellularity. And then there's been some really good advances recently in the genetic side, including recycling of ancient genes to become maybe doing something a little bit new in animals, but then also the emergence and identification of brand new genes, which highlights this idea of genomic novelty. And in a coming lecture here for this unit also, we'll talk about the idea of novelty and look at the importance of novelty as a concept in the origin of organisms in general, but particularly in animals. Now, we're focusing on animals for this class because that's what a vast majority of Evo-Devo has focused on. And to some extent, that may be because as animals ourselves, we're a little bit biased, but it really is because they are an incredibly rich resource with tons and tons of morphological diversity to study, and they're really, really the optimal kind of ideal group of organisms to look at when we are looking at the origin of morphological novelty. Okay, so this is a deceptively simple question here. What is an animal? Now, you might have learned in biology, as I did when I was little, that an animal is a multicellular organism with muscles. And that works for the vast, vast majority of them. But you realize pretty quickly that we leave out some kind of important, even though maybe not as diverse, but some kind of important groups for understanding if it's just that simple. So the definition is a little bit more complex than that. And this paper goes into some of those more complex definitions. I've highlighted them here. I don't really care that you go over them or know them really, really in a lot of detail. I'll look at maybe a little bit more complex, but still not entirely complex definition of animals here in a little bit. Now, animals make up the vast, vast majority of all species on Earth. We've got somewhere like maybe 2 to 2.5 million described species on Earth, and 1.5 million or so of them are classified as animals. So the bulk, the vast majority of genetic diversity, biodiversity, as far as species are concerned, is in the animal group. And even then, it's really biased. Notice that 80% of all animals are arthropods. So a vast majority of animals fall into the arthropod kingdom. And even then, if we broke that down, insects make up a vast majority of arthropods. So there's some groups that are much more biodiverse than others, but others that are important, including mollusks and chordates, us as vertebrates fall into that group. But notice these are much smaller components of the overall animal diversity. So really, if we're talking about diversity of life on Earth, much of it can be accounted for in just the insects. And we're going to use insects as a really important group and as a study group for the lab for this semester also. And then also another important thing to realize that even though we have described, say more than 1 million species of arthropods, even this might be an underestimation because many arthropods are small, limited in distribution, and so they're out there, maybe we just haven't discovered them. it's very unlikely that there are hundreds of thousands of chordate species. Maybe in the maybe fish, there's still a lot of work to be done. But for instance, mammals, we probably describe the vast majority of mammals on Earth with only a few new ones being discovered every year. But arthropods are much harder to describe and discover, much more diverse. So there are many, many more of them. And we estimate as many as 8 to 163. So we don't even know within an order of magnitude how many more are out there. Okay, but the vast majority of that is probably going to be in the arthropod, particularly in the insect group. So just be aware of that. So really, even though 80% of all described, about maybe 70% of all described species on Earth are animals, it might be much closer to 95 or even 98%, depending on how many undescribed species are out there and are still waiting for us to discover them. Okay, so here is our slightly more complex, but still a little bit simplistic definition of what makes an animal. 1, they are multicellular. 2, they have a diversity of cell types, much more so than plants or fungi. Okay, and some dramatically so, some just a little bit more. But so basically, if you want to say what makes an animal, as a very simplistic point of view, it's a multicellular organism with very, very diverse cell types. Okay, and some of those cell types, like the flagellated cell, are unique and distinct just for animals. So there are definitely kind of some cell types that are more important than others when we're talking about animals. All right, so I'm going to talk a little bit about phylogeny, but we've got 2 chapters coming up next week where we'll go into much more depth, maybe review for some of you who have had the evolution class. But I'm expecting a little bit of background about phylogeny and phylogenetic thinking. If you're a little bit lost here, that's okay. Just put a pin in it, wait for a week or so, and you'll get some more background on what is important for phylogeny. But the key here is that in order for us to talk about, understand, study groups of organisms, we really need a phylogenetic tree. It's the roadmap for helping us to understand what we need to look at in different groups and what we need to compare them to. Okay. Now, for a long time, the phylogeny of animals has been it was thought at least to be fairly understood, that we have all the big animals, chordates, mollusks, things that we've been talking about, arthropods. And then we have these more simple animals, the sponges, and their closest relative was the choanoflagellates. And to a large extent, that has been borne out by more recent studies. So it goes in here talking about how molecular studies supported this idea, but made it a little bit more complex. Now, I've already summarized in our last lecture this complexity, so this is a bit of a review, but it's worth knowing. And even this is going to be a bit of a simplification. I've highlighted them here, but for the most part, unless you're an animal geek or maybe a piscicant geek would be a better term, don't worry about the Teratospora and Philasteria, although you need to know that it's a little bit more complex, and there's some small groups that are included within those groups. All you need to know is that animals belong to this large group called the Pisciconta. You'll see it spelled both with the TH and with the T. It's just kind of 2 variants. But the Pisciconta are basically animals and their close relatives, and then the fungi with a few other lesser known groups thrown in. So as long as you know that, you're good. So animals themselves also have undergone a revision. And for a long time, we have understood that the sponges, here's a diagram from Haeckel's Kunstforma der Nature is the German word for it, but it's basically artwork of nature. That both sponges and these ciliated organisms, the comb jellies, aren't important, but for a long time we didn't completely understood the role they played, and that these choanoflagellates, which have these unique collared cells that look similar to cells in animals, including the sponges, were the sister group. And that's largely borne out. Okay, so let's just look at the summary, so I won't get into any more details of that. This is a nice summary of the things you need to know. So metazoa are the animals. Porifera are almost certainly the relative of all the rest of the animals, although there's still some debate about the comb jellies, whether they should be placed as sister group to the porifera or as maybe sister group to all the rest of the animals. So either sponges or comb jellies are the kind of the more ancestral group that branched out before the rest of the animals. And then we have the Cnidara and this small, usually you may not even have heard of it before this class, but the Placozoa. And then we have this big, some people would call it a crown group because this is where most of the diversity is, the Bilateria. Okay? And then, of course, the fungi or the Holomycota, those are really the only important groups, although there are these other unknown. Okay, so just know that, and you should know that the sister group to the Metazoa, as established 100 years ago or so, and is still borne out by molecular work, is the choanoflagellates. Now, although we have defined multicellularity as one of the characteristics of animals, realize that that is not a unique characteristic. In fact, once we look across the phylogeny of all organisms on Earth, particularly in the protozoa, we realize that multicellularity has evolved many times. It's not a unique thing. But there are certain aspects of animal multicellularity, like cellular adhesion and communication between cells, that make it unique and distinct from other types. So we could really say a unique type of multicellularity if we wanted to be very specific. But realize that is not unique to animals. But the second characteristic, they make many different kinds of cells, particularly in chordates like ourselves. Mammals have hundreds and hundreds of different cell types, where most organisms, there's only a handful of cell types. And so we are significantly more complex when we're talking about types of cells than any other organism on Earth. When I say we, I mean both us as mammals, we're more complex than most animals, and animals in general are much more complex based on cell type than any other organisms. So those two are really the best two defining features of what it means to be an animal. And so this transition to multicellularity almost certainly involved some colonial stages, and we see that in our close relatives, the choanoflagellates. So there are probably a number of steps and stages moving up to complete full multicellularity. But again, that's not an uncommon thing. That seems to have happened over and over again in the history of life on Earth. Okay. Now, there's a little bit here, a note about when this occurred that we've already talked about, that the earliest fossils that we can maybe very clearly date to what are animals, although it's maybe still debatable, are about 600 or so million years ago, right? But that genetic studies probably push it back a little bit farther. The earlier ones way farther, and now as we've gotten better and better molecular clock techniques, we're still estimating an origin that's probably a little bit more ancient than the fossil record dictates, which we might expect due to the incomplete nature of the fossil record. Okay, so the last part of the paper, we're going to look at some of the genome and genetic characteristics that originated that are unique to animals. And again, just a summary, we're not going to get bogged down too much in the details. One of the things that made early studies of this complicated was that choanoflagellates seem to have lost many genes. They become kind of genetically simple. And so that led us to overestimate the number of new genes that were present in animals. But once we began to study more and more opisthecons and other groups of organisms, we realized and got a better picture of what it meant genetically to really be an animal. Okay, and so there are two main trends that we see. One is kind of the reworking of old things that were in ancient genomes and kind of a restructuring of the genome. And the other thing were some unique features, unique genes, and I've listed a few here. I don't care that you memorize them, but I've just highlighted them to show that they're kind of unique signaling pathways. So to understand this, so here I've just highlighted here these two things, genome evolution and then the origin of new things. To understand these, we need to understand this concept of homology. And hopefully that's clear in your head. But to establish homology, the best way to do that is to have a clear and well-supported phylogeny. So mapping characteristics onto a phylogeny is the best way to establish homology. Okay, we can do it by looking at the features, making really fine comparisons, but when we map them onto a phylogeny, it becomes readily evident and apparent what things are homologous. And new advances in DNA sequencing and analysis of large data sets has really given us a breakthrough in recent years to allow us to evaluate animals and then compare them to other groups to determine what's homologous. Now, the take-home message here is that the new things, the novel genes, are most likely the product of gene duplication events. We'll reinforce that later in the class, followed by then quick evolutionary development. And there are a little bit over 1,000 novel, HG here stands for homologous groups, but these novel groups of genes that are in animals. Okay, and so these are the key genes to what makes an animal at the genetic level. And it goes into some details about how they determine that, but these have some interesting biological functions like nucleic acid binding, like transcription factors, like genes that bind to DNA, right, and help to turn on and turn off, and then also signaling molecules. And there's a nice table here that goes into some of these things. So these are all transcription factors here. So this is just a list of some of the different ones of that make up novel groups of gene in the animal. So basically, transcription factors, signaling pathways, which many of these are also part of transcription factors also, and then cell adhesion and cell cycle genes. So there's some unique receptors and synaptic, right? So this is with brains, which becomes kind of important here. But so I would maybe put as the most important classes of the new things that came up in animals are transcription factors, including important signaling pathways, cellular adhesion, and then cellular cycle and receptors. Okay. And then also there's an important group I won't get into in detail, but genes that were lost in animals, right? It's not just about what new things you gain, but what old things do you not carry on. And so there's a small group of genes that were lost in the common ancestor of all animals, and so they're no longer important in animals. And then the last thing I'm just going to highlight here, this is kind of an interesting concept. There are two competing hypotheses about the origin of different groups in animals. One is kind of a slow, long, drawn-out process. They call this stew, right? Like, let something stew for a long time and everything happens kind of in a long, drawn-out, slow process. Or the popcorn model, which is rapid bursts of evolution. And after a lot of kind of talk and diagrams, they finally come down to the conclusion that we don't know. A better understanding of rates of gene birth and death and phylogenetic placement of these fossils that we've got that are uncertain is needed before we're able to discriminate whether it was a stew model, long, slow, drawn out, or rapid bursts of new genes in certain groups. Okay. And then I guess the last very conclusion is that there's been, in the last 25 years or so, some dramatic revisions, inclusions of new groups of animals. We've highlighted those. So if you understand, and this is a guided tour to help you understand, you're in fine shape. I don't need you to know all of the details here. I will add some of those important things to the SLOs. And also, if you can do the reading questions for this paper and you feel comfortable answering those two reading questions, that's also a good important sign that you've got what you needed to out of this paper.